Could you tell us what your role looks like?
Yeah, so before going into like my role in particular, let me just give a brief description about how the biotech industry setup is. So we have gene of interest, like the antibody gene that we incorporate it into plasmid, and that we incorporate into a particular cell type. So that is done by a cell line engineering group. They do clone optimization and everything. Once the clone is optimized, they give it to the upstream processing department. Now in upstream processing department, they do a lot of bioreactor based studies. They optimize the cell growth cycle, which media to be used to grow the cells, to grow our protein, to express our protein. Then how long you should keep the cells into bioreactor for them to grow. What is the yield of your protein at each and every day of the growth cycle. Then once that is optimized, all those things are optimized, that cell culture broth, they give it to downstream processing department, where they do all purification steps. They remove cell debris, metabolites, other impurities with 2 or 3 chromatography steps, preparative chromatography steps at large scale, and they purify the protein. And once the purified protein comes, they give it to the formulation development team, where they process it or they buffer exchange it into a desired form which can be marketed. So those things happen at R&D level, and once the process of R&D gets optimized at each and every checkpoints, they finalize this process and then they transfer it to the GMP or manufacturing facility, where they do this at a large scale, the same process. Now I work in analytical development team, where we are a service department for all this. We are a support department for all these departments at upstream, downstream, formulation, development studies. So we develop methods, analytical methods, and then we qualify them. Then we do the method transfer to the manufacturing area and everything. And the thing is, when you develop a particular method, you have to be very precise. I mean, if you are saying that your yield is 98% or your purity is 98%, your protein is 98% pure, then your method should be able to detect that purity level. If there is some impurity that is getting formed, then your method should be able to detect that. And it should be able to detect, and it should be a reproducible method. Means no matter who the analyst is, what HPLC system you are using, at which day of time you are using, if you are analyzing the same sample on different day with different analyst with different system, then results should be reproducible. So we develop those methods, and then we qualify them. We transfer those methods to the manufacturing site, and so a lot of optimization studies and all happen because we provide them the analysis on time. And so I, with my colleagues, we take care of, like, we look after a group of analysts. So we plan their day-to-day activity. We review the results, we give them inputs in their method development process. Then once the results come, we prepare report out of it, and then we ensure that the method transfer happens very smoothly.
Did you always envision in working in biosimilars or as a senior scientist, or is it something that you developed interest once you got into the industry?
After my B.Pharm, I did M.Tech in bioprocess technology, where my second year project was on developing one analytical method to measure the colloidal stability of protein formulation. So from there I was kind of interested in proteins, and because I had worked in a project which was involving protein formulation, I got selected in the interview round, which was happened in Wokhad R&D, where they were working on insulin and analogs. So in insulin analogs, they were making combination therapeutics of 2 biosimilars. So I got to develop the assay and impurity profiling method for that. And because of all these prior background, I got interested into this biotherapeutic field. So I was searching for a PhD in Protein Biophysics somewhere where I can like explore this formulation optimization and other protein stability related thing more. And then I got into the PhD there. And then
But during B.Pharm days, did it sort of boost you or give you the clarity that you want to pursue this in this field, and then you figured it out?
So in my B.Pharm field, in my B.Pharm days, I was interested in protein science a little bit because so the primary question that I had that time was that there are two kind of phenomena: competitive inhibition and non-competitive inhibition. The competitive inhibition was very simple to understand, that you have inhibitor molecule which is binding with more higher affinity than your ligand, and it is blocking the all functional cascade thereafter. But non-competitive was slightly difficult to understand for me because there is a notch where your ligand binds, and there is some random pocket where your inhibitor binds. Once your inhibitor binds, the shape of that notch changes and your ligand cannot bind to it and all. So I was very curious that time that what is happening between these two, like why binding here is changing the structure here, like what is happening there. So that was the initial question that time that I had in mind. Slowly with going into bioprocess technology, and then
Which faculty, as you mentioned, that you had slight interest in competitive and non-competitive. So and our BCP curriculum has these topics. So which faculty was there?
So Krishna ISR used to teach us that biochemistry part of it, and then all this structure activity relationship kind of thing. Professor Ivan Scutino used to teach us. That putting this particular group increases the activity of this particular molecule. Like say for example, if you put halogen at the para position, it increases lipophilicity, and then all those kind of things. So the medicinal chemistry that was taught by Ivan Scutino sir, and then the biochemistry taught by professor.
Okay. And I read that you have done your PhD from IIT Madras. So how was your transition from BCP to IIT Madras? What were the challenges and how did you tackle them?
So the initial thing was that in BCP I had learned many things about pharmaceutics. But then after fourth year, I was just thinking that let's explore the biopharmaceutic part of it also. And people keep telling that biotech is booming. So I thought that pharmaceutic part I have already covered. Let's cover the bio part of it. So I went on doing bioprocess, MTech in bioprocess technology from ICT. And because of my project there, I got interested into this protein biophysics field. So I was just mailing all the professors randomly, whether you have any vacancy for PhD, can I join your lab, like that. So I mailed this Professor Adi Nagnathan from IIT Madras. He said I do have vacancy, but then there is an admission round which you have to clear. Then only you can be a part of my lab as a PhD student. So I said, but then preparing for the exam is not possible when I'm doing a job simultaneously. That time I was working in Wokhad R&D, and like we used to get very tired after this 9 to 6 job, and then I was not able to concentrate on the study. So I just asked him that can you just take me as an intern for 4-5 months where I can like work in your lab, do hands-on gain some hands-on experience with some experiments, and in meantime I can just read about my entrance exam and get prepared for it. So he allowed me that, and because of those 4-5 months that I just I had to risk it. I mean, there was no guarantee that I will make it because that field was also new, like doing B.Pharma and then giving entrance exam in biotechnology in IIT Madras. That was kind of difficult for me, but then in 4-5 months that I prepared, and then I gave entrance exam interview, and then I got selected. So that was the
Amazing.
journey from
That is really
B.Pharm to
impressive.
PhD.
BCP student, there are so many students who are thinking to pursue PhD or get into biotech. So what advices would you give them? What all skills they do need to brush up on, and considering AI and everything. So
Yeah,
what are your suggestions or advice to them?
yeah. So first advice is if you are really interested in doing research or doing PhD, then only you come. A lot of thing is happening in computational biology. The last Nobel Prize that people won, John Jumper and Professor David Baker, they are hardcore computational biologists. So they are into protein design and all. And right now also, like a lot of new biotherapeutics that are coming, they are first screened through this artificial intelligence machine learning tools, and after screening down or after learning those processes through computer, they are doing some experiments. Because just if you start doing experiments, then there will be like a number of possibilities, and you cannot cover everything using experiments. So you have to have this computational knowledge for this to go into research. And most of the people will think that now that is only your field, and they will start giving you job or future opportunities based on your PhD project. So that really defines your future path.
Okay. How would you describe your PhD journey in just one line?
My PhD journey was fun-filled.
Okay.
So because my professor was very helpful, a lot of market has shifted towards biosimilars. So biosimilars means you have to make a similar marketed product, but at the same time your product should not infringe the patent.
Yes.
So you have to use different additives, different excipients, but your product quality should remain same. So those things we take care of as a analytical group in our company. That we develop such methods, such characterization methods, such analytical methods by which we can monitor the quality, critical quality attributes of the molecule.
So basically, you are working with quality control also and biosimilars.
Yeah.
And there its product is the most challenging part of it.
So the challenging part of it is the analytical role we are talking about.
Yeah. So like what working with biosimilars, being in quality control, quality assurance. So that role is a very critical role in any product development.
Yeah.
So then, what do you think is the most challenging part of it, and how you tackle it?
Yeah. So the main challenge that we get here is that since the market is very competitive, many projects are running simultaneously. You have to meet the timelines of each and every project. There are some submission deadlines, then there are some batches planned in manufacturing somewhere, and because of that you have to finish a particular study in R&D way before that. So those things need to be planned properly, then they need to be managed. All the data that is generated should be reviewed thoroughly. So those things are a bit challenging when 2-3 projects are running simultaneously. And since I am into CDMO company, so we have a lot of client projects also, and then maintaining the client deadline, client given, client committed timelines is challenging at times. So those are the main things I find challenging, but exciting at the same time.
Okay. Our students who are interested in working in CDMO, companies, what all skills do you think are very important, and that the students should be prepared, in the graduation,
Yeah.
so that that skills can be, helpful later on.
Yeah. So the first and like the most important skill is not skill you can say, but the attitude of learning new things. I mean, many of the students, they don't have any background of biopharmaceutical industries. They also come to our company as a trainee, as an intern. But if you have a positive attitude, then you can last longer in a company. You can contribute well in each and every project, Then there are these awards giving ceremonies happening periodically in our company. So if you have a right attitude, then from beginning only you can start receiving those awards. You can have that recognition, that name in the company, in various departments and all. So that is one thing. The second thing is, as far as analytical part is concerned, you should, since most of the companies are using HPLC the most, having a knowledge of chromatography and hands-on experience of that is quite beneficial. But though it's not mandatory, because we also understand that in very early stage of your career, you need to be taught things. I mean, it's not like we don't expect people having 3-5 years of experience to join as a trainee or intern. So trainees and interns always have to be in a positive mindset of learning attitude, and that I think solves most of the.
Talking about challenges, is there anything, any skills which BCP taught you and which are helping you in your career?
One lecture by Krishna Priya ma'am that time, and she was just teaching us the significance of washing glasswares. Nothing much. I mean, it was a very basic skill, very like a trivial sort of topic, but she was saying that if you wash your apparatus properly, then you will not get a false results. And if you extrapolate that thing, then you can say that if you do a experiment with proper focus, with everything in line, with all the checkpoints that are covered, then you can eliminate a lot of human errors in the analysis. And in future, if the same analysis happens by someone else, somewhere else, then the results will be reproducible that time also. So that was a very powerful message that I had that time.
So looking back at your college days at BCP, what are your favorite memories in college?
I had very good memories about BCP. So if like one of them was that in the initial days of my B.Pharm, first year B.Pharm, just like it was first month of our B.Pharm, the second year students, the student council members, they had come to our classroom, and they were planning for our freshers party. And the condition for that freshers party was that you have to convince a second year student or your senior to be your date.
Oh.
And then only you are allowed to come into the party. So then, since like we were all very new to this, we took it very seriously, and we all rushed on the same day to find our dates from the second year B.Pharm. And the other one was, it was not a good memory, but now looking back at this time point, it's very like funny. So we had a periodic exams where I think 2 different subjects are covered on the same day with a gap of 3-4 hours or something. So the second exam, the subject, actually I got confused with that, and actually I think it was a pharmaceutical analysis paper on the evening, and I thought it's medicinal chemistry or something. I had prepared for that. So that was a very scary situation that time. So in that 3-4 hours, friends helped me prepare for that pharmaceutical analysis paper. We all sat together, we revised, and then somehow I cleared that exam. So that was quite scary, but it was funny.
Definitely. So what do you miss the most about BCP?
I think all teachers, the lectures, and my friend group. I mean, we are still in touch, but like meeting every day, having fun on daily basis, those things are not happening now. All of them are busy in their career, but we do meet like once in every 2-3 months. That is that.
Which was your favorite subject out of all the subjects?
My favorite subject was biochemistry and then medicinal chemistry.
So both subjects which are taught by Krishna sir.
Krishna and Protein sir. Yeah.
Okay.
So actually it was more like if I like a faculty, I like subject also.
Definitely.
So whenever Ambre ma'am teaches us, we like medicinal chemistry. Whenever Nagasekar ma'am and Mala ma'am teach us, we like pharmaceutics. So it was more like person specific liking of subjects.
Okay. So finally, what is an advice you would like to give to students who want to pursue higher study and research?
So about higher study is that you should select your subject wisely. You should select your lab wisely. For higher studies, your lab, then they should have sufficient ideas on which you can work on, and then you can go through the person's website, the lab website, and you can see 2-3 papers, recent publications, so that you get an idea that currently what research is this lab group is working on. And then if you get interested in that particular thing, then only you should approach them and
So
join.
basically, curiosity is mandatory. So you have to be
Curious and interest.
Yeah,
Your personal
in the
interest.
subject, and then you can push yourself in that.
Yeah.
If a student is confused between industry and academia, what do you think he should pursue first in order to have a good profile, in order to have good experience, exposure?
So what I would suggest is that having some industrial exposure early in your career would help, and then simultaneously you can prepare for the research, academia and all, because you are already into academia, and then from bachelor's to master's and PhD is you will be in academia only, so you don't know about industry, how industry looks like. So spending few years in industry will help you decide whether to pursue industry or to go into academia. Because if you don't have any knowledge about something, then you cannot judge whether to go into industry ahead or into academia. So having some initial years of your careers in industry would definitely help. But then again, at the end of your industrial exposure, you should have that SWOT analysis done. You should have pros and cons of academia and industry both. Like say, having industrial experience in early career helps in deciding this.
And one last question is, we have World Pharmacist Day coming up. Any message to our BCP student who are the future pharmacists?
We are the people who make medicines. So we have to make medicine in such a way that we should think that what if this medicine goes inside the body of our dear ones. So we should have that quality of the medicine that should be maintained all the time. So that keeping that thing in mind, we should work into our respective positions where we are currently. I mean, many of the alumni are at very big posts in different industries. So, but this thing should be kept in mind to this younger generation also, and to the people who are already working in industry, that we should maintain the quality of the pharmaceutical product that is going into the market all the time. So there should not be any compromise in that.
You correctly mentioned about not compromising on quality. It aligns with our BCP's motto, our vision of college. We have always made sure that our students do not get compromised in academia. And now that we have also other student-led activities, we are developing a holistic approach towards this curriculum. Something which is very much needed, in today's era.
So recently I have seen a lot of posts coming from BCP, and I'm quite happy that we are these things what we couldn't do in our time. Now this new committee, new student body is involved in, so that's making me very much happy.
Thank you so much, sir, for sharing your journey with us.
Thanks,
and
sir.
Thank you for accepting our request and coming to college. I hope you like the change, and it was.
So it was a very nice initiative. Thanks for calling us. I visited the campus after I think 13-14 years, so it was quite refreshing for me. I met faculties here, and so nice to see all positive changes here.
Thank you.
And I wish to be in connect with BCP current body and other alumni through this platform.
Definitely. And BCP is your home, so you
Yeah.
are always welcome here.
Thank you. Thanks.